everyone, and it's really nice to meet you for those who don't know me. My name is Sabrina Hardin, and I'm a research assistant under the PI, Dr. Brian P. Herman, and we're with the Neuroscience Developmental and Regenerative Biology Department at the University of Texas at San Antonio. I'm really excited to show you my project on determining the function of ID4 ERT2 through expression profiling and functional characterization. So before I begin, I would like everyone to take a moment to visualize a happy couple, happy loving couple who dreams of starting a family. They dream of little footsteps running in the hallway and the joys of parenthood. However, their dream is not being realized due to infertility, which impacts millions and millions of individuals throughout the globe. So, human life and humanity as we know it is one whole consisting of two halves. So human beings are diploid organisms, which means we get half of our genetic information from mom and our other half from dad. And so infertility experienced by any of these parties is detrimental to fertilization and essentially creating that human life. And so as we know, males contribute to half of that genetic information through the process of fertilization. However, 9% of males all over the globe experience infertility at some point in their lifetime. And those who are experiencing this infertility and do want to go into like family planning and creating human life, getting those and receiving those treatments, they on average spend about 18% of their entire annual income just on treatments alone, and that does not include major surgeries like tissue biopsies. And so averagely on a normal day, every day, men produce over 80 million sperm. And so that is a lot. And that is through the process of spermatogenesis, where a primary spermatocyte goes through 2 rounds of meiosis to create a haploid spermatid, which then differentiates into these flagellated sperm cells that we're more familiar with. That flagella helps them go through the fallopian tubes, and they create a zygote. So as you can tell, that spermatogenesis is critical to maintaining male fertility throughout their lifetime. And spermatogenesis is backed by SSCs, which stand for spermatogonial stem cells. And so stem cells throughout biology are known for their regenerative properties. And in this case, spermatogonial stem cells have 2 functions. One is to auto-renew, as a stem cell does, where they regenerate their own populations of SSCs to maintain spermatogenesis throughout a male's lifetime. And they can differentiate. So they can begin the process right here. They can either auto-renew or differentiate and start that process of meiotic division to develop mature sperm cells. This is a cross-section of a seminiferous tubule within the testes. And this is where you can kind of see these SSCs going through their different developmental stages and where they go through elongation and into the vesicle to be stored within the testes. However, my specific project, as you saw in the title, is highly specified, and it's one minor cog in a very large machine. And so the bigger picture are theories of SSC specification. So when we're born, as soon as we are created as a zygote, we are given primordial germ cells. And so within males, they develop into spermatogonia. During birth, they can either start the first wave of spermatogenesis, or they can create the first population of SSCs, which can then auto-renew, create a bigger population throughout the lifetime, and create steady-state spermatogenesis. However, we don't have a lot of information regarding the biological methods and mechanisms which helps determine them, why they either go to the first wave, why they make SSCs. Determining why and how SSCs function and are created can lead to profound impacts on fertilization treatments, and on the flip side, potential contraceptives. So a graduate student in my lab, Dr. Roa de la Cruz, because she got her PhD very recently, she developed 2 theories with my PI, Dr. Herman. One was determination, where it's these prospermatogonia, since they were developed from those primordial germ cells, they had a clear path throughout their life. They either start the first wave spermatogenesis, or they have that steady state spermatogenesis by creating SSCs. That's it, clear path, there's no altering that at all. But there's also another theory where maybe these spermatogonia choose at random. Understanding why and how, again, that these SSCs in particular are created is something that's critical to fertility development. So the way that we can kind of trace this, and the way that my lab is approaching this, is through two means: lineage ablation as well as lineage tracing. When we are doing these lineage ablation, we are creating or knocking out a certain gene so that it's no longer expressed. And so if it's no longer expressed and it has some implication on a lack of sperm, then we can kind of determine if it's a determination or selection. And there's also another form, which is lineage tracing, where we put a biomarker so we can see if these mature sperm have a biomarker. But as I said, there are certain genes and mechanisms that we don't necessarily understand what role they play. And so my lab is looking at a whole bunch: DDX4, SOX3, IDTA, and one of them that I'm working on is ID4. So ID in ID4 stands for inhibitor of DNA binding, which activates and regulates the expression of certain genes. Feng Sun in the University of Shanghai of Biosciences in 2015 did some studies regarding ID4 positive and also ablating these ID4 cells. They determined that ID4 positive cells within the testes function as normal. So they create a steady state SSC population and they continue to produce sperm. However, when you knock out that gene or deactivate it, it creates a much smaller testes, as you can see here with the red arrow, a much lower weight of the testes, and as you can see here, I showed you a healthy cross-section of these microtubules where the SACs go through their process, and these are the ablated cells. So as you can see, it has a profound impact on the SSC population, which is detrimental to creating that sperm. And so now we understand that this is a gene of interest. We can potentially use this as a marker for SSCs throughout that prospermatogonial stage. The way that we can do that with the lineage tracing and ablation is through the use of a Cree driver, which is a genetically engineered tool where you can, essentially it's a tyrosine recombinase, so it's an enzyme that can essentially induce recombination in the genome at a specific site. So Cree ERT2 is a tamoxifen inducible Cree in particular, where recombination can occur at the LOXP sites, which are represented here by these arrows, when tamoxifen is present. So here you can induce, you can prevent it from being expressed if you attach it to our genome, or our gene of interest, which is the Cree driver. And these are the two coding exons. Attach it right there. And then that's when we can manipulate the usage of ID4 in our model organisms. It is also an ortholog, the ID4 gene, and in this case, an ortholog represents a gene that has similar functionality between species, and this would be in humans and in mice. However, like I said, I am a very specialized, specific cog in this giant machine, and so my goal, my research goal, is when we're creating this transgene of ID4 with Cree in order for us to manipulate it in model species, we are creating a genetically modified gene. And so when we're creating a genetically modified gene, we need to validate that it's functioning as ID4 would. The way that we can do this is through expression profiling. Is it being expressed in the tissues it should naturally? So expression profiling methods, this is where we essentially are getting certain tissues. The reason why we're using these tissues and RNA specific is because every cell has DNA. Every cell has that recipe book. But when you differentiate into different tissues, like let's say the colon and the brain, you are specifically having those promoters with that RNA so that they can develop into different tissues. So it's a small recipe in a very large recipe book. We are using 12 adult mice and 12 P6 mice, which are postnatal day 6. So these are tiny little pinkies. They are 6 days old, but they're very relevant in regards to developmental, because they're going through a very critical stage in when their organs are differentiating and they're developing into adults. So we use 6 males and 6 females. 3 of them are ID4 positive, and 3 of them are not ID4 positive. So we extract 24 tissues throughout the different components, so the lower half, the thoracic cavity, and essentially the brain. And so we go through RNA isolation, RNA quantification by creating that RNA template. We treat it with DNA so that we don't have any of that big recipe book within our specific recipe. And we do a reverse transcriptase to develop a complementary DNA strand so that we have that DNA to be amplified by the PCR machine, which is the polychain reaction machine, which amplifies that DNA over and over and over so we can see banding to see if that gene is being expressed. So our expected findings, since we have not done this yet, is we expect to have banding in our PCR machine within the testes, the epididymis, and the colon. Because as we know, just generally without the transgenated gene, this is where ID4 is found. That's just how we know through RNA sequencing in general. This is another piece of a project that I've worked on with DDX4. This is an example of what our PCR could look like. This is just the GAP-DH results, which just show that we do have the complementary DNA. This is also credited to Indy Ubo, which is a post-bac here at the lab that I've been working with. This DDX4 shows banding on the testes and ovary, which does confirm expression profiling, as we know that DDX4 is only found in the testes and ovaries. Like I said right here, we do expect banding to be in the testes, the epididymis, and colon. However, that is a work in progress as we develop these mice and they grow up into adults. The future direction, we're going to continue mRNA profiling with both the adult and P6 mice, continue genotyping just to make sure that they are ID4 positive mice, and laxi staining just to confirm, because with science we just like to prove our proof. Our future projects, we want to utilize this transgene as a marker for SSC specification, so we can find out more about determination, selection, and potentially develop some treatments and maybe contraceptives on the flip side. These are my references, and I would like to thank my PI, Dr. Herman, as well as my mentor, Dr. Jordan, as well as the data provided by Lorena Roa de la Cruz, as well as Indy, and my McNair Scholars Program, and my cohort. And this is just some grant information. So this work was supported by the NIH grant R01HG90007 to Brian D. Herman. And the transgenic mice were genetically modified using WSU Gene Editing Reagent Core with Dr. Lizette Madison and WSU Animal Production Core with Dr. Miranda Bernhardt. And that is it. And I will take any questions.